Before we get started, I have a quick favor. I've been self-funding the Finding Genius podcast for 5 years now. I've done over 3,000 episodes. And as you can see on YouTube, we're up over 1 million views on the channel, which is fantastic. The next thing I really want to push on is to get up to 10,000 subscribers. Because once we do, we'll be able to put a donate button and we'll be able to solicit donations to help keep the podcast running and to also get the Finding Genius Foundation moving along. We have a big project studying anxiety, depression, and PTSD and working on a product to help people overcome these problems because I've seen them explode recently after the last 2 years of the whole virus situation. So if you would, please subscribe to the podcast. That would help us tremendously. Give us a thumbs up and check in the description for Buy Me a Coffee. It's about $5. If you could buy me a coffee, I'd really appreciate it. It would help keep the channel going, and I love coffee. Thank you.
Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
Go above and beyond.
they become very good at what they do, but only
0.1%.
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
With Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast, now part of the Finding Genius Foundation. My guest today is Courtney Stairs. She's an associate senior lecturer, part of the microbiology group at Lund University. And we're going to talk about what's called microbial eukaryotes, or protists, and her research surrounding them. So welcome, Courtney.
Thank you so much.
If you would, maybe a quick background on yourself, how you got to this area, and then why do you study protists? What's interesting about them to you?
Yeah, thanks. So protists are, I guess, kind of like a biologist's playground because there's so much unknown about these organisms. So, for example, maybe I'll just step back a second and explain what a protist is, because I think perhaps it's not a
Sure.
usual dinner time conversation. So if you were to think of all of life on Earth as this sort of tree, like maybe something you have in your front lawn, a maple tree or something, and if you were to think of every leaf on that branch as a major group of organisms, animals, plants, fungi, each one of those, so these things that you see around the world today, each one of those would maybe represent one leaf each. And then every other leaf on that tree is represented by microbial life. And this is fascinating to think of how much out there makes up a vast majority of the biomass on our planet is completely unexplored. And so that's what I'm interested in. So microbial eukaryotes are essentially anything out there that is not a plant, animal, fungi, or bacteria, if that makes sense.
So what are some examples of microbial prokaryotes instead of eukaryotes?
Yeah, so prokaryotes are things like bacteria or archaea. So these are things that, like E. coli, maybe you've heard of the scary pathogens like E. coli or chlamydia, or these sort of, things that cause harm to us. But some examples of microbial eukaryotes that can be harmful include things like the malaria pathogen, for instance. So malaria is a disease that kills millions of people, and this is caused by a single cell organism that can be transferred between insects to humans. And there's other nasty microbial eukaryotes out there.
What are the ones you focus on? Because I'm sure there's a lot. You can't do everything.
Yeah, exactly. So the ones that we work on is those that live in areas that don't have oxygen. And that's kind of what fascinates me. So I'm not so focused on what the organisms are, but more like how they can survive in these different environments. So we study organisms that live without oxygen, which sounds kind of crazy, right? Because life as we know it, we need oxygen. You know, if you hold your breath too long, life is not very fun. But for some organisms in different environments on our planet, they need to live in these environments. It's a completely uncolonized niche that they can thrive in. And if they figure out how to do that, then they don't need to fight with the rest of us.
What do they use for metabolism then? Methane, sulfur, what and what?
Exactly. So if we're happy to go into chemistry, then I can try my best to explain this. But basically, it's easier to think about why do we need oxygen? So when we think of our metabolism, the reason we need oxygen to survive really fundamentally, generally comes to electrons. So in order for our metabolism to move forward, we need to figure out a way to get rid of our electrons, which sounds kind of crazy. But basically, we eat sugars. We metabolize those sugars into fundamentally electrons and some water and carbon dioxide. And in order for us to move that metabolism forward, we have to get rid of those electrons. So we use oxygen. It's a great electron acceptor. It loves getting electrons. So there's some organisms out there that they metabolize sugar, they make electrons, they make CO2, but they have no oxygen because they're living in, let's say, our gut or the bottom of the ocean. So they have to find an alternative suitable electron acceptor. And that's where this research is interesting, I think, because instead of using oxygen, they pop their electrons onto protons. And if you think back to first year chemistry, when you add some electrons and some protons, you get hydrogen gas. And so this hydrogen gas can become the fuel for other microbes in the community. So you get this sort of like metabolic cooperation. One organism, it needs to digest these sugars, it needs to get rid of its electrons, it puts them onto protons to produce hydrogen gas, which is then consumed by another organism.
What does that mean then, that anaerobic organisms will tend to be in the same locale or partner with aerobic ones?
They could partner with aerobic ones. So you get this really neat situation where you can have organisms that love to live, like specialists that say they love to live without oxygen. And if there's ever oxygen there, they're going to quickly move away. And then you get these sort of generalists that could maybe flop between the two communities. And these are the critical organisms to really understand how you can shuttle biomass from one zone, let's say one completely anoxic zone, unto another, because you can get these organisms that can be permissive in both environments. And this is how we can funnel biomass, electrons, what have you, between different communities.
Well, again, what organisms are you studying and where? I mean, like in our guts, a lot of it would be anaerobic in there, especially in our intestine, small intestine.
Exactly.
I'm sure there's a lot of microbes there. People don't even know what's in there. But again, which ones are you studying and where?
So we study organisms that live mostly in the natural environment. So these are things that live in intertidal zones or, in the Baltic Sea, where a lot of the oxygen has been completely removed from the Baltic Sea owing to a lot of human activity, unfortunately. And so you get these anoxic zones. And so the organisms we work on, as I said, I'm kind of not a specific organism, but we work on things called breviate protists. So these are small, like 5 microns, think like super, super tiny organisms that can eat bacteria, for instance, and they can shuttle this, as I was saying, these electrons from various processes to prokaryotic organisms, which can then do other metabolisms such as methanogenesis, making sulfides or nitrogen.
Okay, well, again, so these anaerobic bacteria and other organisms, they usually feed off hydrogen gas, or what about methane or sulfur or other things?
Yeah, so that's a really good question. So it's really a microbial soup in some of these environments. So they will, one organism might make some hydrogen, and then another organism could take that hydrogen and use it for other processes. So it could make methane, it could make hydrogen sulfide, or it could make things like nitrogen or nitrous oxide, which those gases are not super great for our environment. So for instance, methane, we know, is a powerful greenhouse gas. Sulfide can really destroy aquatic communities, and nitrous is another harmful greenhouse gas. And these sort of byproducts of metabolism can have profound impact on the more cuddly creatures that live in these environments, such as fish and algae.
Well, again, but you're talking about anaerobic organisms, so how are they supposed to be in the presence of aerobic ones? The environment would be different, I would think, right?
Yeah, but so you can have these, but this is what's so cool about microbes is, sure, you'll have these super specialists that if they ever see oxygen, they'll die or run away. But then it's the ones that live in the middle, the ones that can do both the anaerobic metabolism, so help to create the methane or the sulfides, and that can transition into these more little bit of oxygen environments that are potentially.
Well, wait. So are there anaerobic organisms that can exist in an aerobic environment, but they just process in an anaerobic way? And do you have the converse of that?
Yeah. So I mean, things aren't, I mean, as much as we'd like to say things are anaerobic and aerobic in sort of this black and white perspective, I think it's easier for me to see it as a spectrum. So there's these so-called oxyclines, which is a fancy way of saying a gradient of oxygen in various environments. So you don't have this sort of border that's just like, okay, from this, in this millimeter, it is normal atmospheric oxygen. And then as soon as we cross over, it's completely anoxic. You get, in fact, this gradient. And in these organisms that live in the middle, we call like microaerophiles or facultative anaerobes, which are fancy ways of saying like flexible organisms. They can kind of have a foot in both ponds, if you will.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now, back to the show.
Okay, I didn't realize that. You're right. I thought it was just a black and white division of the two. But then I thought, how do you transition from one environment to another? And, what's considered an environment? And, right, anaerobic creatures, do they live again in a totally isolated anaerobic environment? There's an envelope and then it passes to an aerobic environment, but it's not like that. Like you said, it's a continuum.
It's a continuum in some environments. Yeah, and the ecologists call it an oxycline or a redoxycline. Yeah.
So if you look at an oxycline and you go from the aerobic to the anaerobic side, what does it look like? Are there microbes that just live in the middle of the two? And then when you get to something being totally anaerobic, they're all gone and now you're only left with purely anaerobic ones? Or what would it look like if you profiled it?
Yeah, that's exactly the case. And folks have done a lot of really cool studies where they've examined what the community looks like along an oxycline, and you can see the types of organisms that are there. So by that I mean the taxonomy or the species relationships of the organisms in the different environments. That changes, but fundamentally what changes is what those organisms can do. So the organisms maybe at the more oxic zones, they're doing oxygen-dependent metabolism, and then the ones that are at the very end of the oxycline, so the purely anaerobic environment, they're doing a completely different metabolism. So it's this nice, almost stratified community.
But what about the metabolites formed? Do they build up and then cause any problem for the organisms and reset the boundaries of, a microenvironment, let's say?
Yeah, that's a really good point. So, nature has found very clever ways to get over this. So this idea of metabolic cooperation, of where one organism will produce a potentially harmful byproduct that is then quickly consumed by another, is prevalent in these environments. So you can get a byproduct such as sulfide that could be reoxidized to something less harmful by another organism in the community. So you get this sort of symbiotic for the common good situation.
Okay. So what kind of experimental questions are you trying to answer? What are some of your setups? What are you looking into?
Yeah, so when we're trying to understand, really at the basics of this sort of metabolic cooperation, how do organisms communicate? So, think in the natural world, you have these two organisms. One needs to make a metabolite that's then consumed by something else. How do they find each other? Because in the lab, of course, we can put the two organisms in the same tube and then great. That's they're going to find each other just because I've given them very limited space. But what happens in the natural world? How are these organisms finding each other? Do they need to communicate? Do they send signaling molecules out that would maybe tell them, hey, partner, can you come over here and help me with my metabolism? So we're trying to understand that. And then we're trying to understand, do they need to touch each other? So for instance, if you want to send out your hydrogen, it's going to quickly diffuse away, but you really want someone to help you consume that hydrogen because it helps your metabolism move forward if you can form these nice, stable relationships. So we want to understand how do organisms control who they're sharing their metabolites with. And so basically to do that, we're trying to recapitulate it in the lab as much as possible and trying to see, like, do they need to touch each other to share metabolites?
So like in an organ, you get different cells that have different functions, yet they're all part of the same organ. In biofilms, I guess, I don't know, but I guess you would see probably differences in as you go through a biofilm. One side where the top layer is, facing oxygen, the bottom layer is anaerobic.
Yep.
I would think you would see like a gradation of, of bacteria types within a biofilm and different,
For sure.
metagenomics and et cetera. Is that what's been observed?
Yeah, that's what people are doing. It's exactly the metagenomics. It's a great point. Like, this is how people are trying to get at this question, at least from a predictive point of view. So if you can see who in a biofilm can do what, then you can kind of predict how they are interacting in space. But what we're trying to get at is taking these predictions that metagenomics has really afforded us and like actually testing it in the lab and seeing what metabolites are being shared between organisms to try to validate all of our metagenomic predictions.
Well, what if you had I'm imagining a setup. Let's say you have a biofilm and, the top part of it is in a more aerobic environment. The bottom's anaerobic. And you run it for a while, you do metagenomics, and you manually flip it. So now all of a sudden the top part is in the you take the biofilm off and flip it around, What do you think would happen? And would it adjust or would it die? Would you see like a fast changeover of epigenetics in the bacteria and their jobs would suddenly change? Like, what do you imagine would go on?
Yeah, I mean, I'd have to run the experiment, of course, to even begin to think about it. But if I would guess that, biofilms can be pretty fluid. So the easiest thing would be everything would just flip, because I think the organisms that maybe prefer the anoxic lifestyle, they're constantly sensing oxygen, and if oxygen's there, they might be moving away from it. So I would actually expect the biofilm to just go back to normal, like go back to how the original setup was. The cells would just change place. But I don't know. Maybe that experiment's been done. I don't know about it.
Yeah, because I'm thinking about, let's say the microbiome of people, and I always wonder what happens in the border zones between one microenvironment and another. What lives there? What like, look, if you look at your skin, the outer layer of cells, they're exposed to the air and just all kinds of stuff. And then as you get 1, 2, 3 layers in, those cells aren't exposed to nearly as much. And as your skin sloughs off, the cells below now migrate to the surface, so they would have to change as they see this new environment. So I wonder if that would be maybe I don't know, maybe that would be a good...
it's an excellent question. Yeah. At least in the gut, there's been some work showing how different microbes can sense the oxygen, and they use that as a way to tell if they are close to the epithelium. So, for instance, there's some gut pathogens, at least from the eukaryotic perspective, so the protists, the ones that I know of, when you, in the lab, expose them to a little bit of oxygen, they're anaerobes, they don't like it, but if you expose them to a little bit of oxygen, it actually induces pathogenicity factors. So those sort of machinery that is important for attachment to the epithelium or invasion of the epithelium. So oxygen can be harmful to these anaerobes, but sometimes it can also be a signaling molecule, like tells the parasite, hey, I'm getting close to the epithelium. Now I need to start evading the immune system. I need to start attacking the epithelium so that I can get to where I want to go, if that makes sense.
Yeah, it's really interesting. I guess they're doing a lot of chemotaxis. They're following their own gradients and seeing what's coming in, what's going out, et cetera.
Exactly. Yeah.
Is there, I guess there probably is also not necessarily quorum sensing, but like threshold sensing on the amount of oxygen where you see like all of a sudden a change in metabolism of these bacteria, if up or down.
100%. Yeah. This is exactly what they use. It's an important signaling molecule. It's an important metabolic molecule. So yeah, oxygen it has a lot of power.
Very interesting. What other research questions are you trying to answer in this regard? Like, what other interesting behavior are you evaluating or looking at?
Yeah, the understanding how eukaryotic cells, so these are, as I said, basically everything on the planet that's not a bacterium, plant, animal, or fungus. So this is a vast majority of the biomass or a significant. Let me rephrase that because I don't want to misspeak. So if we zoom out a bit and just think of biomass of organisms on our planet, animals account for approximately like 2 gigatons of carbon, which is a lot. But then the protist contribution to the planet is 4 gigatons. So there's twice as much biomass of protists as there are animals on the planet. And of those microbial eukaryotes, there's tons that are anaerobic. And what's kind of cool about these protists is that some have evolved to produce hydrogen gas. Some have evolved to use alternative electron acceptors, such as nitrates and fumarate and other metabolites. And it's just fascinating to think that various lineages have independently adapted to live without oxygen, and they found different strategies. And so we're trying to understand what are all the different strategies that these organisms can use? How important are they for the environment? Are there new strategies out there that we haven't discovered yet? And so right now we're really focusing in on the hydrogen generation, as I said, and then also different electron transferring strategies.
Well, you know, some people are using bacteria or algae or whatever to, you know, separate water or to produce electricity or to digest stuff, whatever. Is anyone modeling the pathways? Like, can they be understood, you know, aerobic versus anaerobic organisms, or, you know, semi-aerobic, ones that use methane or ones that use nitrogen? Like, are all these pathways being modeled, and how complex are they? Can they be replicated without these organisms, or you really need the organisms to do it?
That's a good question. Yeah. So I think synthetic biology, I've been thinking about starting to dabble in it just to see if, so this is this idea that we can engineer systems that can perform a metabolism that we desire. So for instance, as you said, like anaerobic bioreactors that we could maybe harness from yeast, or maybe photosynthetic bioreactors using algae. And I think what's so attractive about looking to nature is that, you know, nature's had a head start. They've been doing this. These microbes and protists in particular potentially have been doing this for 2 billion years. So they've kind of perfected the art. And I would think that if we study some of these organisms, we can really start to take advantage of them, to maybe learn from them. How have they learned to, as you say, like use methane or produce methane? And can we kind of take advantage of that and maybe pop it into a synthetic system?
Well,
I think that could potentially be the future.
Yeah. What other, I don't know, compounds have you seen organisms living off of? Are there any novel ones that are like incredibly rare?
I mean, not in my line of research. I don't think I do anything that cool.
I don't know if you've seen anything or heard anything in the literature. Like, I'm sure some people work on like the black smokers down at the bottom of the ocean, and I guess those things live on sulfur. And, you know, they're probably a more exotic version of what you're looking at. But
yeah, exactly.
Okay. Are you looking into any commercial applications of these, or is it more basic research type stuff?
It's at its core, I do fundamentally basic research. I have ideas. So some of the, hydrogen generation is, of course, the bread and butter for these sort of organisms, because it is fundamental to their metabolism, and they need to figure out clever ways to make hydrogen. And of course, on the converse, there's organisms out there that have clever ways to consume that hydrogen. So we have kind of stumbled upon a way to make hydrogen that is maybe energetically more favorable than the normal strategy. And so we're trying to investigate that a bit more, but it's really early days, so I don't have much to say beyond that.
Well, since hydrogen is such a big part of what goes on, are you able to look at, deuterium in place of hydrogen, or environments where you have significantly more, deuterated atoms or water than non? Because that could affect the system probably pretty dramatically.
So you mean naturally occurring deuterium, or do you?
Yeah, like in some
eating in
right, naturally occurring. No, naturally occurring. Like in some environments, let's say organisms using hydrogen, they're consuming it. But maybe this particular environment is, I'm just making this up totally. There's like one.
Because I'm like, I've never heard of natural deuterium, like at an amount that one could detect.
Oh, really? Yeah, I would think different amounts, even if it's very small. And I just wonder if that affects the metabolism of these organisms. Is there any natural environments where deuterium is, present at 3 times the level as others, and therefore it affects their metabolism?
I'm not sure. So like deuterium is basically hydrogen with an extra neutron, right?
Right, exactly, yes.
Yeah.
Yep. Yeah, that's okay. I just wondered.
Yeah, no, I don't know. At least from my my very electron centric point of view, I don't see how that would affect the metabolism. But yeah, sorry,
Okay.
don't know.
Yeah, I guess another thing that comes to mind too is what about phage activity in aerobic versus anaerobic versus these middle of the road organisms? Has anyone even looked and studied that? And do you get unusual types of phages, for anaerobic bacteria versus aerobic?
Ooh, that's a good question. Yeah, it's an excellent question. I don't know if anyone's thought to look along the spectrum of physiology. So I think that's hitting a dark spot in my knowledge. But I can say that perhaps like viruses and phages are grossly underestimated player in these communities. They are responsible for microbial turnover at a significant rate. So it could be that if there would be differential lysis activity. Yeah, I don't know. I don't really want to comment on that. But good point.
Yeah, it's pure speculation. But yeah,
I have no idea. Yeah.
Okay. Are there any environments in which radioactivity plays a role? Naturally occurring. Let's say there's anaerobic organisms and rocks that they contain uranium, and therefore there's significant radioactive activity, and other organisms that feed off of that you run into.
I mean, there are plenty of organisms that can survive in these sort of environments. The relationship to oxygen is not super in my wheelhouse of these guys. Sorry, no, that's
okay. No, it's okay.
Yeah.
I'm just asking all the the craziest organisms on Earth. Where would they be? So going back to fundamentals, are there fundamental differences that you see versus aerobic versus anaerobic, or again middle of the road organisms like when they continue on these oxyclines again? Are they fundamentally different, the anaerobic versus aerobic bacteria, or not really?
Yeah. So I guess like in addition to having different metabolic pathways, they have different like chemistry within their cells. So basically in our cells and in oxic cells, like aerobic cells, you kind of have to protect your iron. If your iron is exposed to oxygen, it can form a harmful byproduct in this thing called the Fenton reaction, which is just a fancy way of saying a bunch of radicals are forms that can cause damage to your cells. So when we're respiring oxygen, like we need oxygen, obviously, but there's harmful byproducts from that. And so our cells have many strategies to protect against this. But if you're an anaerobe and you probably never see oxygen, you have fewer of these sort of iron defense systems. So a fundamental difference between aerobic and anaerobic organisms just on the inside is what the iron looks like. So that's kind of cool, and that's a difference between these organisms. But there's different metabolism we have in our cells where we need oxygen beyond just, making energy. We need oxygen to make important cofactors in our cells. So for instance, hemoglobin requires oxygen to be synthesized. But if you're an anaerobe and you maybe need heme, there's strategies to make heme that have evolved independent of oxygen. So you see the exact same molecule being produced in aerobic and anaerobic cells, but the way in which they produce that is completely different. And I think that's just fundamentally like fascinating that nature has found two different ways to make the same molecule. And this is because the chemistry is different in these two environments.
Okay. Any other fundamental differences? Like do anaerobic bacteria and other organisms tend to have slower metabolisms? Do they divide more slowly, move more slowly? Anything that's in common?
Yeah, I mean, it's hard to... The coolest thing about nature is that it's so diverse. There are fast-growing anaerobes and slow-growing aerobes. So it's actually quite nice that there's no general trend that one can put across these organisms. So there's... Yeah. Which is actually unexpected because, like, energetically, you would think that anaerobes would be slower, but that's not always the case.
Okay. Well, very good. Where can people keep tabs on your research and find out, what you're working on? Where can they go?
Yeah, so they can, check us out on X, formerly known as Twitter, and then also at our homepage, thelabupstairs.online.
Well, I got the lab upstairs. That's cool.
Yeah.
Well, very good. Well, Courtney, thank you for coming on the podcast, and thank you for being tolerant of my, random walks here and crazy
No,
questions.
it was great. I mean, it's really fun because like you bring up points that I have never even considered, and it makes me want to go back to basics and try to find those answers.
All you need is hundreds of millions more dollars in research, and you can figure everything out. That's all.
That's
Easy.
right. Yeah, easy peasy.
Well, excellent.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.